All right, let's dive into a really clever metabolic trick that's happening inside your red blood cells as we speak. Seriously, it's the key to how your body makes sure oxygen gets exactly where it needs to go. It's called the Rapoport-Leubering Cycle, and it is fascinating. Have you ever wondered how your body manages to adapt when you're up in the mountains with thin air? Or how it pushes through a really intense workout? Well, the secret isn't just about breathing harder. It's actually a super sophisticated biochemical tweak happening inside your red blood cells, a process that puts oxygen delivery above everything else. So let's break down exactly how that works. So here's our game plan. We'll start with a basic problem, the oxygen delivery challenge. Then we're going to zoom right into the red blood cell itself. We'll explore a unique little detour in its metabolism, the glycolysis bypass, and the crucial molecule it produces. Then we'll see the clever energy sacrifice the cell makes. And finally, wrap it all up with why this stuff is so important in medicine. Okay, first things first. Let's set the stage. Our body's relationship with oxygen is basically a constant logistical puzzle. It's not just about grabbing the oxygen, it's about delivering it to the right place at the right time. Think of hemoglobin like a fleet of delivery trucks. It's amazing at picking up cargo, that's the oxygen, in the lungs. But the whole mission is a failure if the truck just drives around with a full load, right? It has to be able to unload that oxygen at its final destination. Your muscles, your brain. That release mechanism is every bit as critical as the pickup. So where does all this oxygen delivery magic happen? Well, for that, we have to look inside the delivery truck itself, the red blood cell, or erythrocyte. And this isn't just any old cell. It's a highly specialized courier with some really unique quirks. Red blood cells are fascinating mostly for what they don't have. They have no mitochondria. Those are the powerhouses of the cell, so they can't burn fuel using oxygen. This means they are completely 100% dependent on a single pathway for all their energy, glycolysis. That's their one and only metabolic highway. But here's where it gets really cool. The red blood cell has built a special bypass, kind of like a detour off that main glycolytic highway. And this special little pathway is called the Rapoport-Leubering Cycle, or sometimes just the Rapoport-Leubering Shunt. And get this, it's not some tiny, insignificant side road. A pretty huge chunk of the glucose in a red blood cell, we're talking about 15 to 25%, is deliberately sent down this shunt. So that begs the question, why would the cell divert a full quarter of its precious fuel supply? The pathway itself is actually beautifully simple. It starts with a molecule called 1,3-bisphosphoglycerate, which gets pulled off the main glycolysis path. Then it gets converted into a really unique molecule, 2,3-bisphosphoglycerate, which we'll call 2,3-BPG. And finally, that molecule is changed back into 3-phosphoglycerate, which just hops right back onto the main glycolysis highway like nothing ever happened. Now, that first critical step, creating the 2,3-BPG, is handled by a key enzyme called Bisphosphoglycerate mutase. You can think of this enzyme as the traffic controller that's waving cars off the highway and onto the scenic bypass route. And the enzyme that directs traffic back onto the main highway? That's 2,3-bisphosphoglycerate phosphatase. What's wild is that both of these functions are actually part of the very same protein, managing the whole cycle from start to finish. So now that we know how this bypass works, let's get to the most important question of all. Why does it even exist? The entire reason for this whole detour, this whole cycle, it all comes down to the function of that one special molecule, 2,3-BPG. And this comparison right here explains everything. 2,3-BPG is basically the key that tells hemoglobin, hey, let go of the oxygen. Without it, hemoglobin has a super high affinity for oxygen. It holds onto it really, really tightly. But when 2,3-BPG is around, it binds to hemoglobin and lowers its affinity for oxygen, making it way easier for that precious cargo to be unloaded in the tissues that are desperate for it. So it makes total sense that our bodies crank up the production of 2,3-BPG whenever we're under oxygen stress. At high altitude, where there's just less oxygen available, or in conditions like anemia, the body makes more 2,3-BPG. Why? To make sure that every single molecule of oxygen that is picked up gets delivered as efficiently as possible. It's just a fantastic adaptive mechanism. But there's a catch. This incredibly useful function, it comes at a price. The Rapoport-Leubering cycle isn't free. There's a metabolic trade-off, a sacrifice that the red blood cell has to make. See, back on the main glycolysis highway, that direct step from 1,3-BPG to 3-phosphoglycerate, that's an energy-generating step. An enzyme just plucks off a phosphate group and uses it to make a molecule of ATP. It's a direct energy payout for the cell. The Rapoport-Leubering bypass completely skips that ATP-generating step. By taking this detour to make 2,3-BPG, the cell sacrifices that energy payout. The net gain from this part of the pathway is a big, fat zero. For every molecule that goes down this path, the cell is willingly giving up potential energy. And our source material, Satyanarayana's Biochemistry, sums this up perfectly. It calls it a way to dissipate or waste the energy not needed by erythrocytes. The red blood cell's main mission isn't making energy for itself. It's transporting oxygen for the entire body. So it happily sacrifices some of its own energy efficiency to be a better oxygen deliverer. Okay, so this elegant piece of biochemistry isn't just something to memorize for an exam. Understanding this cycle has real-world clinical implications, especially when things go wrong somewhere else in the glycolytic pathway. Check out this table. It shows how defects elsewhere in glycolysis can have a massive ripple effect on our cycle. For instance, if you have a defect early in the pathway, like with hexokinase, you make fewer intermediates downstream. That leads to low 2,3-BPG, which makes hemoglobin cling to oxygen too tightly. But on the flip side, if you have a defect late in the pathway, like with pyruvate kinase, everything upstream gets backed up. That means more precursor for 2,3-BPG, so its levels go up and hemoglobin unloads oxygen very, very easily. All right, so quick quiz to make sure we've got this down. The enzyme that kicks off this entire shunt, the one that makes our star molecule 2,3-BPG, what is it? That's right, it's Bisphosphoglycerate mutase, the gatekeeper to this whole process. And let's just reinforce the big picture here one more time. The whole reason 2,3-BPG levels shoot up at high altitude or during anemia is because it's an adaptation, right? It forces hemoglobin to be better at its job, releasing oxygen to help tissues that are desperate for it. And that really brings us to the ultimate conclusion here. The Rapoport-Leubering cycle is a master regulator. It gives the body a way to fine-tune oxygen delivery based on its moment-to-moment needs, all without wrecking the red blood cell's own metabolism. And really, that's the main takeaway. The cycle is just a perfect example of a biological trade-off. The red blood cell gives up a tiny bit of energy for itself in order to do a much, much better job of serving the entire body. It's just an incredibly elegant and efficient solution to one of life's most fundamental problems.